Buenos días, me llamo profesora Dedee Murrell y estoy una jefe y profesora de dermatología en Sydney, Australia, en la universidad y en el Hospital San Jorge. Y voy a continuar en inglés porque es más fácil para mí. The subject of today's lecture is new treatments for difficult to manage autoimmune blistering diseases. These are my conflicts of interest. I had a wonderful holiday with my husband in Cuba in January 2023, where I had the pleasure of having dinner with Dr. Olen Grelovio. What wonderful hospitality and friendship. And we enjoyed very much exploring your country over a 2-week period and would love to return sometime. This is St. George Hospital, where I work, and these are some of Sydney's beautiful beaches, nearly as pretty as Varadero, and our beautiful harbour near where I live. Today's topic will be about biologic targets for pemphigus and pemphigoid. How can we measure blistering diseases in clinical studies? What treatments do we have approved so far? What trials are going on for potential newer and safer treatments? Thanks to research that has been going on the last 30 years, especially in blistering diseases, we know a lot about the pathogenic abnormalities in these diseases, and a lot of monoclonal antibodies and other small molecules have been developed which can block these pathways. So we're going to have a look at these in the two main blistering diseases, pemphigus and bullous pemphigoid. But before we can do that, we need to talk about simple things like how we as dermatologists describe these diseases. When I first began to look at this back in 2005, we found that the papers had so many different descriptions or definitions and ways of measuring the severity of these diseases or outcome measures that it was impossible to pull them together. So despite all the sophisticated science that had been going on, together with my friend, Professor Victoria Worth at the University of Pennsylvania, and the charity for patients with blistering diseases, the Pemphigus Foundation, we tried to persuade a lot of the doctors who were busy competing against each other in labs to start working together to talk one language, to describe these patients and have one set of outcome measures so that we could proceed with clinical studies. These are the initial experts who joined our group in this endeavor, meeting in Paris in 2005 to start discussing in early morning meetings around the world when we were attending different conferences to try and gain consensus. 3 years later, this first consensus paper was published in the Journal of the American Academy of Dermatology, which describes these different stages, early endpoints and late endpoints for clinical trials, and definitions of what a complete remission means, a partial remission means, minimal therapy of prednisone means. These are the types of simple definitions. For example, in green here, control of disease activity means the time at which the new lesions have stopped forming and the established lesions have begun to heal. In order to be sure that the patient had reached that stage, at least 2 weeks later, 80% of those lesions had to be healed, and that meant that the prednisone or other steroid could begin a slow taper. At the end of that taper, the ideal would be in green to have complete remission of steroid therapy, being the absence of new or established lesions while the patient is off systemic therapy, meaning steroids, for at least 2 months. Or they could be still on minimal therapy, defined as less than or equal to 10 milligrams per day of prednisone or half an adjuvant therapy. Or they could have a partial remission. And what is a flare? It's the appearance of 3 or more new lesions a month that are not healing spontaneously within a week, or by the extension of established lesions in a patient who achieved disease control but not remission yet. The next goal was to establish a disease extent measure for pemphigus, because you need a scientific way to score any disease, and because dermatology is visual, that needs to be a reliable visual score, and it has to have good inter-rater and intra-rater reliability and correlate with other measures of disease severity. It has to show responsiveness so that you can see the score changing in clinical trials. And so that was our next goal, is to discuss what needed to be included in that trial, to review the literature, and to come up with an instrument that would be reliable between different raters. At that time, the German group were establishing a score called ABSCIS using the rule of nines from Burns, and we began the PDI development as an international group using the principles that had been adopted for lupus scoring with the CLASI instrument, whereby you have scores for the skin, scores for the mucous membranes, and you don't score scarring or damage as activity because it's not easily reversed. This is the ABSCIS score, which has quite large percentages in it. The smallest is 1%. That's okay when you have big areas of involvement, but it becomes more difficult when you just have a tiny blisters estimating the size. It also only scores the oral mucosa and asks questions about eating a Western diet. The PDI, on the other hand, picks out the top 12 areas of the skin that are involved and doesn't count lesions. It just jumps up in stages as to less than 3 or more than 3 lesions in each site, which is easy to notice immediately, and only count them if there are less than 3. Otherwise, they go up only if the size of the biggest lesion gets bigger. It also scores if damage is present or not, to remind you not to score old lesions as activity. The scalp is a common area in pemphigus that requires weighting, and it's divided into quadrants or the whole scalp. And 12 of the commonest involved mucosal sites are also scored in a similar manner. This takes 3 to 4 minutes to accomplish once you're used to doing it. The next was to test these scores prior to publication, and that was done at the University of Pennsylvania and published in the JID. And here you see how it compares with other scores in dermatology. This is a Cosmin analysis. Each score should score well on each of these 7 criteria. And you see the PDI score at the top. Everywhere in green, it does a good job. In red, it hasn't been done yet. In yellow, it's not so good. If you look at the PASI score for psoriasis, you can see that it was used before it was validated and that many elements of it are not good or haven't been validated. A somewhat better situation with the EASY score for eczema. The PDI score, since this chart was used, has now got the responsiveness and MCIDs accomplished in the last year with our group. And if you look at the CLASI instrument for lupus and the CDASI for dermatomyositis, they both perform very well, like the PDI does for pemphigus. Our group then developed and validated a score for blistering diseases quality of life called the ABQOL, and one for the quality of life when you're under treatment, the TABQOL. Each of these is no, a little, a lot, and an awful lot, scoring out of 3 with worse outcomes. So it's out of 51, 17 questions, and it's been validated and published in Australia, then the restudied in the US, and published in the BJD, and then it's been revalidated in many different languages and cultures, such that it's in use for clinical trials. Another instrument is how to quantify the toxicity from steroids, and I did this in collaboration with other medical colleagues, led by John Stone at Harvard University. And we recently published both our own study and more recently an international collaborative study of using the GTI in our clinics. So now to the treatments. What do we have in the toolbox? Well, we have these things that suppress and destroy B cells by attacking the CD20 antigen expressed on B cells. and rituximab and ofatumumab are the two main examples. Rituximab has been around for quite a while now and is used for lymphomas and many autoimmune blistering diseases and other autoimmune diseases. Within days, the B cells are gone, but because the plasma cells will still live on for another 3 months, you have continuation of production of autoantibodies. And it usually returns to normal levels at 9 to 12 months. The first study was an independent study by the French group led by Pascal Joly and published in The Lancet. And it was a randomized controlled trial of rituximab and short duration prednisone versus the traditional high standard dose of prednisone for pemphigus vulgaris. For severe cases, the prednisone dose was 1 to 1.5 milligram per kilo per day, and for moderate pemphigus, 0.5 to 1 milligram per kilogram per day. There's a schematic I can show you that shows you better. You see here at the top, with these blue triangles, the big standard doses for the severe patients. You see there, they got 1.5 milligram per kilogram per day. Moderate got 1 milligram per day tapered over 12 versus 18 months. And they were randomized against the experimental group, who, in addition to 2 infusions of rituximab, 1,000 milligrams 2 weeks apart, and repeated after 12 months, then 18 months, the primary endpoint of complete remission off steroids was read at 24 months. In this group, you see that they had half the doses or much lower doses of prednisone and only for 3 months or 6 months. Over here, what you see, the red line is the B cell counts, which in the placebo comparison group were high, and in the rituximab group were suppressed. So in the trial, 90 patients were randomized half-half, and at 2 years, the complete remissions off therapy were almost 90% in the rituximab arm versus 34% in the standard treatment arm. The complete cumulative duration of complete remission off steroids was 7 times longer with rituximab, 446 days versus 62 days of the standard treatment. The cumulative prednisone dose was 1/3 in the rituximab arm with half the number of side effects. So that was a very good, promising outcome and led to the registration of rituximab by the FDA. And it was the first line rituximab that should be used rather than waiting for the patients to have a lot of relapses. It's more effective than corticosteroids, has a major steroid-sparing effect and fewer side effects. The Roche company also sponsored a trial of rituximab plus prednisone plus placebo mycophenolate mofetil randomized against placebo rituximab and actual MMF and steroids, and they chose a 1-year endpoint. They essentially got the same results as the other study, but after 1 year. So another CD20 molecule is ofatumumab, which was initially delivered by infusion, also used for lymphomas, but now available as a subcutaneous treatment. And I designed a trial of that, and we had launched it and enrolled 37 patients in this phase 3 trial when the drug was purchased by another company, and the trial was shut down to develop this for multiple sclerosis. But I have used this off-label for some pemphigus patients with stubborn lesions, where you can use subcutaneous injections, and they're given every month, so they don't leave the patient immunosuppressed as long as rituximab. And we're just about reporting those outcomes. Another promising molecule is the C5a receptor expressed on mast cells, which are involved in bullous pemphigoid. And in mice, at least, one of these MAP kinase inhibitors avoided having bullous pemphigoid expressed in the mice. They also improved with sodium chromoglycate, which is a mast cell stabilizer. Bullous pemphigoid often involves the expression of IgE. And in this particular study by Janet Fairley, she found that they're high in 40% of BP patients and almost 70% of MMP patients. but as the only expressed type of IgE, it's only 5 to 8% in these diseases. It's more frequent in urticarial BP. We know that there is an inhibitor of IgE high affinity receptor called omalizumab, and there have been lots and lots of studies and case reports of omalizumab working for BP, but the company doesn't seem interested in sponsoring any trials on it. There was an early study of infliximab, but it only had a small number of patients and didn't reach statistical significance. Then the next molecule to be tried was a Bruton tyrosine kinase inhibitor, borrowed again from hematology, which blocks a lot of the innate immune system and the adaptive immune system. So you see that these BTK inhibitors do the same things as JAK inhibitors and as rituximab and as steroids, but don't have the off-target effects of steroids. I designed this study in dogs with pemphigus at the University of California in Sacramento vet school. These dogs just had the tablets only of the BTK inhibitor, and they got better very quickly. Their PDI scores decreased, and they came off therapy and relapsed and were retreated. We then developed a molecule that was bioavailable in humans called PRN-1008 and tested it in a phase 2 trial published here in the British Journal of Dermatology. Here you see the PDI scores over the first 12 weeks in dark blue, and you see the percentage of people that went into control of disease activity. 50% or 52% achieved control of disease activity on this, plus up to 20 milligrams per day low-dose prednisone within 4 weeks, and it continued to increase up to 12 weeks. Some patients even went into complete remission by 12 weeks, and then the drug was stopped, and patients continued to improve. Here are the PDI scores, I beg your pardon, in dark blue and pale blue, and you see that they're gradually decreasing, and the kids' steroid dose requirement decreases as well. And here you see the antibody levels against desmoglein 3 going down. Here you see the chance of going into remissions increases with time. And here you see the gradient of reductions of the BDI score in the study, where 4 of the patients were only on the BTK inhibitor and not any steroids at all. And the dose of steroids was only 0.2 milligram per kilo per day, compared with the rituximab studies I showed you, where they were on 0.5 to 1.5 milligram per kilo per day. We then designed this phase 3 randomized controlled trial of PRN-1008, 400 milligrams twice a day, randomized against placebo with a standard corticosteroid taper after disease control. The primary endpoint was to be a complete remission on minimal therapy, defined as 5 milligrams of prednisone per day at 37 weeks. So they had to be like that for at least 2 months prior, and various secondary endpoints. Unfortunately, this drug was also sold off, and Big Pharma changed the endpoints to 10 milligrams of prednisone, and that was too high. A lot of patients in placebo arm can get better with 10 milligrams of prednisone per day, and it missed its p-value. But with the original outcome measures, if they'd been retained, it was successful, but it's not going forward, unfortunately, for registration for pemphigus. Next comes an interesting class of drugs called FcRn inhibitors. This system in the body is shown there in blue, and it's a way of recycling antibodies. Antibodies are taken up into cells, into endosomes, and if they are bound to the FcRns in the membrane of the endosome, then they don't go into the lysosome for destruction. They're recycled back into the blood. So if you block these FcRns, as shown with the red dots, then you can push more antibodies into the dustbin lysosome and reduce quickly the level of antibodies. So this is the mechanism. It's being used for and registered for myasthenia gravis and transient hemolytic of the newborn in babies. And there have been 2 trials going on, one in pemphigus and one in pemphigoid. The phase 2 proof of concept for efgartigimod in pemphigus vulgaris was conducted in Europe to look at dosing and 10 milligrams per kilo of the patient intravenous initially, but subsequently subcutaneous dosing on a weekly basis rapidly reduced the number of immunoglobulin levels within 6 weeks, certainly by 8 weeks, and you see there non-controlled phase 2 study with the antibody and levels going down. So let's see what happened when this drug was used. In pemphigus, it didn't reach its primary endpoint, unfortunately. I don't know if that's dose-related or not. In bullous pemphigoid, we've gone through the same process with even more experts interested, published outcome measures, created and validated a score called the BPDAI, which is shown here. It has erosions and blisters. It also has urticaria erythema, if that's the main manifestation of the patient. And in this disease, we know there are different types of immune system involved in different diseases, and that BP is a type 2 disease with certain cells in the innate immune response and others in the adaptive immune response. In the type 2 inflammation, the main players of cytokines are interleukin 4 and 13, IL-5, IL-31, IL-25, TSLP, and IL-33. The type 2 inflammatory response is mainly directed against fighting helminthic parasites. We know that these cytokines have multiple critical roles in bullous pemphigoid, which are shown here, that lead to blister formation and itch. And we know how this interplays between Th2 cells producing interleukin 1, leading to itch, basophils producing IL-4 and 13, leading to itch, and eosinophils IL-31, leading to itch. But what would happen if the IL-4/13 part of it were blocked? Would the patient's not only itch improve, would the disease improve? Another reason to choose this as a target is that they're also released by mast cells, which are playing a role in bullous pemphigoid. We also know that patients with BP are twice as likely to develop serious infections as controls, and that many die due to infection. So if something other than steroids, which have been shown to be the strongest risk factor for these infections and deaths, wouldn't it be fantastic? So the primary outcome in these trials has been complete remission on minimal or no therapy, and secondary outcome measures these scores. These are the trials that have been ongoing so far for bullous pemphigoid, which I'll spend a few moments explaining. I've been involved in the design and conduct of all of these studies. So the first one is of efgartigimod, the FcRn inhibitor that we just described, and the trial is called the BALLAD trial. As you know, the pemphigus equivalent trial just failed to meet its primary endpoint, but bullous pemphigoid is a different disease. There was a similar trial design in that the patients at baseline, after confirming diagnosis, are randomized to receive efgartigimod subcutaneously once a week, plus oral steroids, whilst the placebo group is getting the placebo efgartigimod. The primary endpoint of the proportion of participants in complete remission while off all steroids greater than 8 weeks is being assessed at week 36, and then they have a 52-week follow-up. And this is still ongoing and hasn't yet been assessed. The other trial I also have been designing since 2017, and which is just read out its week 36 results, is the Liberty BP study. This one was randomizing dupilumab, standard dosing, subcutaneous every 2 weeks versus placebo, in addition to standard steroid tapering. The patients are supposed to taper off steroids ideally by week 16. It's quite a rapid taper, and then see if they relapse more often in the placebo arm than in the dupilumab arm, with the assessment occurring at week 36. These results were recently read out and reported and are very exciting. This study did meet its primary endpoint with a significant p-value, with 20% of the patients sustaining complete disease remission who were on dupilumab versus only 4% of those who weren't. The components of this primary endpoint are complete remission off steroids by week 16. That wasn't significant. Absent, probably because the tapers were the same at that stage. Absence of rescue therapy during treatment period, much better in the dupilumab group. Absence of disease relapse after tapering the steroids, again, much better in the dupilumab group. You also see statistical significance for multiple secondary endpoints, including 90% reduction of disease severity, clinically meaningful itch, reduction in disease severity from baseline, reduction in itch from baseline, and days of complete remission with no steroids. So this interim results looking extremely promising. There's another readout at week 52 that we're waiting for. So very, very encouraging. In fact, there's a growing body of real-world evidence showing clinical response with targeted biologic therapies in bullous pemphigoid and pemphigus. There's dupilumab, anti-IL-4/13 for BP, omalizumab, anti-IgE for BP, and rituximab, mainly for pemphigus, but in some recalcitrant BP cases, it has been used. So, in conclusion, we have some very well validated objective scores to measure pemphigus, the p-dye and the abscess, and the BP-dye and the abscess for bullous pemphigoid. Our consensus definitions for primary and secondary endpoints have been designed to assess the ability of new treatments to produce outcomes that minimize or avoid steroids completely. We have well-validated quality of life tools for these studies, the ABQOL and the TABQOL, in many languages and cultures. And nowadays, there are many sponsored clinical trials of biologics happening in blistering diseases at last. Rituximab is the first FDA-approved biologic for pemphigus, but I predict that we're going to be seeing more very soon. If you'd like to learn more about these diseases, which are fascinating, I'd like to encourage you to have a look at these two books. I'd like to acknowledge the input of global experts in these rare blistering diseases who are shown here, working as part of our team. And I'd like to thank you for inviting me to join you there in Cuba just for this little short presentation. And buenos días y muchísimas gracias de invitarme. And here I am in Sydney, heading up to summer. Thank you.